Hello and welcome to this part of the lecture Cell and Gene Therapy. My name is Marius Rosenberg and I'm going to present a part about the commercialization of a life science technology and how to get an idea into the market. So I'm a founder of a life science company. I started a spin-off out of the University of Aachen in 2012, 2013 called Adhesys Medical, where I was the co-founder and the CEO of the company. We sold the company to a pharmaceutical group in 2017, and then I remained there as a manager for another couple of years. Next to that, I'm also an investor in life science companies, first and foremost startups that I support also as a mentor. And I'm also a venture partner in a venture capital fund called 10x Founders. So I'm a little bit all over the place in the startup scene. And I hope within this lecture, I can give you an insight into the possibility of commercializing a technology through the means of a startup. So this lecture is cut in 3 parts that I will present today. The first part is really giving you an inspiration why you should commercialize your research and the technology. And then I will tell you the story of my own company, Adhesys Medical, just to give you an example of how such a story can actually go in real life. The second part of the lecture is going to be about the preparation of a startup. And here I've chosen 2 main topics. Of course, founding a startup has multi-dimensions and is a highly complex business actually. But most questions that I get from students and researchers in the early phase is about can I actually finance a company? And then, of course, how do I really commercialize the life science technology? So I will focus on that. This will be the largest part of this lecture. And then the last part will be about getting started. And here I will focus, actually, I call it on the art of pitching, because the very first step of commercializing a technology is that you have to pitch it to somebody, to an investor, or even to your colleagues and co-founders. And it is something that is not really taught at university. So I will talk a little bit about that in the third part. So let's get started here. And first, I would like to give you a little teaser why you as a scientist should actually consider commercializing your research. And I will start with a sort of easy example that brings this closer to your mind. So over the last years, we have seen a lot of innovation in the world. And if you just look at these people and these covers of magazines, we can see that software has really changed our world as we know it in the last 25 years, I would say. And many aspects of our life have been changed dramatically, and productivity has increased, and a lot of sort of challenges and problems we had in the last decades were solved by these software innovations. But now we are facing a different challenge, and this is here the example of climate change. This challenge, in my opinion, cannot be solved by software alone. So if we talk about climate change, if we talk about the generation of electricity, if we talk about CO2 emissions, these things can only be changed if we change the hardware, the underlying hardware. And if you look at the 25 years that I just mentioned about software, and at the same time, while there was so much innovation and productivity increase in the software space, at the same time, our emissions for our planet have continued to increase over that time. The solutions that we need, the hardware solutions, have not yet come to pass. So as a scientist, I know most of you are in the field of life sciences, but life sciences, and we've seen it in the COVID pandemic, have the same challenges. And so the question is, what can we do about this? And I believe that that innovation can come more specifically from startups. And if we talk about hardware innovation, that is sort of in the startup world called deep tech innovation. And deep tech innovation, in my opinion, is really the only way of tackling these challenges. And I will talk a little bit about deep tech in the remaining part of the lecture. So I've here a slide about the question, what is deep tech and what does it differentiate from other innovations? So deep tech is always based on an innovation, on an invention that is proprietary, IP protected, that comes out of a scientific or technological breakthrough. And it has always a hardware component to it. So if we think about also other software enabled innovations like AI, those can also be called deep tech because they have a very, very hard hardware component to it. So you've seen it over the last couple of years. AI was only made possible by huge advancement in chip generations, and actually we will see that to continue. But there are also, of course, other fields, and especially in the life science space, it's about genomics, it's about synthetic biology, and it's about all aspects of biotech. And so what is the difference between a software innovation and a deep tech innovation? So it really comes to sort of the market side of things. And so if you look at software, so software as a service, SaaS, here you can make revenue basically from day one, because you can start selling your product to customers. You will not make any revenue on day one, of course, but the revenue can increase over time, and then you have the famous hockey stick effect in later years. But with deep tech, it's different. You first have to develop the technology to a final point before you can bring it to the market. And most specifically in life science, actually, you're not allowed to sell a technology before you have tested it thoroughly on animals and on humans, and which is actually a good thing. So you cannot make revenues in the first years, and then once you bring the product to the market, ideally you have sort of this jump up, and then you can start generating revenues. So again, looking at the startup space, I would like to explain a little bit what sort of different cycles of innovation we have seen. And if you look back over the last, again, I would say 15, 20 years, we've seen a first wave of innovation in the B2C space. This is sort of business to customer space, where companies like in Germany, I've used German examples here, like Zalando, marketplaces, but internationally Amazon, have sort of changed the way how we do shopping, directly from the business to the consumer. And then there was a second wave that started in the B2B space, where software was made for other business, for other companies like Personio, HR software like Celonis and others. And that was sort of the dominated of the startup space over the last decade. And now deep tech is actually, from my perspective, the next wave. And what differentiates it from the previous waves is that it's more technology based. So it can be B2B2B. So you're actually not directly selling to a business customer. You might be selling your technology to another business who will integrate it into other technology and then sell it to another business. So the depth of the technological integration is even more deep. And here are some examples, and I've picked actually some that are closely related to RWTH to make this more tangible, that are on this route and that are doing sort of these deep tech innovations. And now coming back to life science, which is the area that you are most familiar with. So life science is actually the oldest and most established field in deep tech, because it started when we look back really at the biotech revolutions in the 1970s with now really well-established players like Genentech or Tanox, who have been founded in that time. And why is it sort of the first field of deep tech? Well, one combining factor in venture capital or in the startup scene is that those companies need a high amount of capital, while they have no revenue or low revenue, as I explained it before. And so venture capital is needed. Venture capital plays a significant role. And so actually life science companies were the first beneficiaries of the very early venture capital funds that were started in the 1970s. And so for life science, as I explained before, like it's true for all deep tech fields, the technological risk is greater than the market risk. So you first have to really develop the technology to a final stage before you can commercialize. And sort of the challenge of convincing the customer to use your product or to buy your product, like Amazon or others really had, or Zalando, if you remember Zalando in the early days, for them it was really hard to convince people to buy shoes on the internet. That is sort of a market risk. But if you look at life science and deep tech companies, the biggest challenge at the beginning is the technology. But life science has also some very particular advantages and challenges. So the advantage is, for example, that there is high regulation, which might sound like a challenge, but it actually gives you planability. So because there are regulators in the US, there's the FDA, and in Europe, there's the EMA, or there are notified bodies for medical devices, it gives you a little bit of planability. You know exactly what you have to do, and you can plan your capital needs actually according to these rules. And then there are a lot of established players, large pharmaceuticals group like Johnson & Johnson, for example, who have a lot of capital. And so for startups, it's actually highly relevant that there is a so-called exit pathway, so that there are companies in the market who in the end will be interested in buying your company. And so this is a big advantage in the field of life sciences, because these players exist, and in some other technological fields they may not exist. And then there's also sort of a clarity about the value creation in life science. So when you pass certain thresholds in the development of your product, for example, the design freeze or the achievement of preclinical or clinical data, you have very clear value inflection points of your development that you can present to investors, which then allows you to raise the next round of venture capital. In other fields, you may not have those clear value inflection points or milestones. And then also in life science, you have a low need of personnel in the early phase, because the development of a medical device or a pharmaceutical product, you can actually do with limited amount of people, because you don't need a large sales force, for example. But there are also, of course, some challenges. So the market access is highly complex with well-established players in the market. And for many products, it is very hard to sell a single product alone, because it's typical for the industry to have so-called bundles of product that the players sell to the hospitals in a larger bidding process. And so that's sort of the problem of being a one-trick pony, like a pony that can only do one trick, that this may not be enough. So it's always important to partner with other companies. And then, of course, there's the binary outcome of studies that you also know of. You've heard out of the press that some pharmaceutical product has failed in a class 3 trial, which means that basically millions or sometimes billions of dollars of investment are lost in such a study. And another aspect is that in life science, it's rather rare that you see the so-called unicorns or decacorns, so very, very large companies. And there were some rare exceptions like BioNTech in Germany, for example, who were really successful with their COVID vaccine. But it's very hard to do basically the market access, building up the sales force, and really creating a new company there. So it's rather typical for life science companies to be sold in earlier stages. So again, also as an inspiration, if you think about your career in life science, and if you want some inspiration in which areas you should also specialize in your studies, so I would recommend you to think about sort of the larger trends in society that you can focus on if you want to develop solutions that can help patients. And so one bucket is, of course, to anticipate the changes in society, may it be age-related or behavior-related, or induced by society, and develop solutions for that. The second bucket is about basically fighting things that can really kill us. And so that can be nature-inflicted, like plagues like malaria or also COVID, but it can also be self-inflicted, like antibiotic resistance, which will be a huge problem over the next decades. And so as humanity, we are, of course, aware of these problems, and there's a high willingness, of course, to invest a lot of resources to prevent this from happening. And I can encourage you to look into that as well. And then sort of the last bucket is more opportunistic. look at what the big pharma companies are doing, or especially what they're not doing anymore. So a lot of big pharma companies have actually reduced their own R&D, and they're focusing on very specific fields. So it's very worthwhile to look at sort of the empty fields, the white spaces that the big pharma companies are not addressing anymore, and develop solutions there, because again, those are all companies that like to acquire new technologies, and they will always be on the lookout for good startups in these fields. So that was the first part of the inspiration, more on a general level, why you as a scientist should commercialize. And I truly believe that if you are a scientist, I truly admire scientific breakthroughs and scientific success, but I also believe that it should not stop with a publication or a patent. I think sort of the greatest revelation of scientific success is really to bring it to the market, to bring it to the bedside, to bring it to the patient, and that can be done through startups. So I hope that some of you will choose that path as well. So to give you a little bit more inspiration, I would like to talk about the story of my own startup and how it came all about. Also showing you some pictures of what it really looks like. But let me start quickly explaining what Adhesys Medical is doing. The pitch is really that Adhesys Medical is in the field of closing wounds. So basically a field where we have used ancient technologies for a very long time, like stitching or stapling or cauterization. And if you look at it, there hasn't been really much of innovation in that field for a long time. But there is still a problem there, because if you look at the numbers, there are still about in half of the organ and cardiac surgeries, there's a so-called bleeding-related complication, which means that during the surgery, the surgeon encounters a problem where he or she has to interrupt the surgery to stop a bleeding. And actually, if this bleeding is severe, we have a mortality rate of up to 20% in the OR. So there is really still a significant problem when it comes to bleeding. And this problem actually even gets bigger if you look at trauma patients, so who have multiple uncontrolled bleedings, and there the mortality rate is even higher. And while this is a big, I would say, human catastrophe, it also has a huge economic downside for the hospital, because the incremental cost of fixing a bleeding-related complication can cost the hospital more than the actual previous intervention that was being done. So if you look at it from a I would say pure economic side, you have a medical problem that needs solving, and you also have an economic challenges for the healthcare system and for the hospital. So if you put those two things together, there is an opportunity for delivering a solution. And so when we looked into the problem, when we were developing this product, we were thinking about, okay, what sort of characteristics does the perfect surgical adhesive needs to have? And from our perspective, there were four properties that we need to address. So one is that the product needs to be usable in a wet space. So if you operate within the body, there's always fluid, may it blood or others that are around. So the product needs to work with that. At the same time, you also need a so-called strong and an elastic seal. The product needs to be very strong, but at the same time elastic. So it needs to move. If you have moving parts like vessels, organs, if the product is too rigid, it will break open. So it needs to be strong at the same time, but also flexible. And then also it needs to be, or it should be, fully synthetic. Why? Because there are a lot of biological products out there which serve as glues or hemostats, but those are biologically based, and that can cause infection or allergic reaction with the patient. So this is actually a problem. And then the fourth category is that it needs to be biodegradable, because everything you put inside the body, at some point it needs to go out, and so at some point it needs to be biodegradable. So with that in mind, we developed two product lines. One is a topical skin adhesive, which can be used on the skin to close so-called topical wounds. It's a class 2 device regulated in Europe. And then we have developed a whole string of products that can be used inside the body for gastrointestinal surgery or cardiovascular surgery or hernia surgery. And that, of course, is a class 3 device. I will talk a little bit more about that later in the lecture, what that means. But it's highly regulated because it's in the body, it's biodegradable, it doesn't come off, for example, off the skin, and so it has to adhere to higher standards. And to give you a little bit the story of the company in a nutshell, so we started as a team out of RWTH Aachen University in 2012, 2013. We incorporated the company, and it was developed in a collaboration between a chemical company and the university. And at some point, the chemical company wanted to withdraw from the project, and then there was the opportunity for us to take it over. And I co-founded the company together with another colleague from the chair of entrepreneurship that I was working with at that time, and a surgeon from the university hospital, and a chemist also from the chemical company that co-developed the product. So we started in 2013, then really as a GmbH. We founded a company and acquired basically the patents that were needed for the technology from the chemical company, and put them into our newly founded GmbH. And then we needed, of course, funding. I will talk about funding in a little bit more later, so I will not dive deeper. So we acquired the first funding in 2013, and then also in 2014, and then started building the company. And basically on day one, it was just me working full time on this technology, and my background is in business, and so I had to look for other for other employees who could also fill in the technical gaps. So we built out a team in the first years. At the same time, we also wanted to challenge our approach and the technology. So we went to business plan competitions, and we actually won the RISE Business Plan competition, which is the largest business plan competition for university spin-offs at that time. And we actually won more than €1 million in prize, or $1 million in prize money during that competition. We were the first non-US startup to win that competition. So this gave us a big boost and also allowed us to expand into the United States. And we opened our first office in the US 2015, did our first clinical study also in 2015. And then we were also able to get the ISO certificate as a medical device manufacturer. That's ISO 13485. I will also talk about this a little bit more later. And were then sort of allowed to produce and sell medical devices. We then also entered into the first collaborations with industry players, and signed distribution agreements. And then in 2017, we were fully acquired by the company Grünenthal, which is a pharmaceutical company, with whom we then continued the development. So at that time, we didn't stop, and I also remained on board. So we continued the development. We continued to do clinical and preclinical studies, and then continued and finished the development of the product. So, and just to explain it a little bit to you, how it all went down with some pictures. So just showing you what the office looks like. So the very first office just looked like this. So it was, just one desk and 2-3 people who we could fit in this room. We soon then expanded into, again, still one office. We could fit I think up to 6 people in there. And then a few years later, we were then finally in a larger office with our own lab space where we could do some lab work. We actually outsourced most of the production, but we could do some lab work in our office as well. So on the business side, we also had some business milestones that I would like to show. So when we won the RISE Business Plan competition, actually we were allowed to ring the the closing bell at Nasdaq in New York, which means so when you ring the opening bell, you go public with your company. If you ring the closing bell, it's just a marketing stunt, but still we could get our picture portrayed at Times Square, which was really nice. And you see the picture outside of the iconic Nasdaq building at Times Square, and there was in the Nasdaq, it was me and my co-founder Alex standing next to me, and Malte Brettel, who is a professor here at RWTH, who supported us very strongly during the entire route. He was there as well. So that was the moment when we then received our ISO certificate, so the ISO 13485. And that was a moment to celebrate for us because it really established us as a company. And that was the moment when we submitted our first product to the notified body for product approval. So in the end, you have to sign sort of the technical documentation. I will talk about that later as well. And all the responsible people have to sign sort of this cover page. This is when we signed the cover page. We submitted, I think, more than 10 sort of paper binders for approval, but that was just the final page we signed. That's how the product looked like when it was done. So from research to sort of like final approval, it was a route of a couple of years, but this is how it looked like when it was all done. That was the moment when we signed our first licensing agreement with a pharmaceutical company, and that was then the moment when we actually sold Adhesys Medical to the Grünthal Group in 2017. That was the view into the Notary Public office when you sign all the paperwork, so that was the life cycle of a startup, and hopefully I gave you a little bit of inspiration how it could look like. Now we come to the actual part of doing it, of working through it, and so I will start with financing. Because financing is a very important aspect of building a startup. And I often get the question, because I talk so much about venture capital, is it the only option? And I want to start with that. So venture capital is highly relevant for financing startups because it's sometimes the largest source of capital you can tap into. But there are a lot of other ways of financing as well. So, and I'm listing them here on the right-hand side. I'm not going to read them all, but you can bootstrap, which means that you sort of try to finance with your own money for some time. You can get different kinds of loans or grants. Use the money from business angels or friends and family. I will talk a little bit about that. So there are other ways of doing it. But venture capital is highly relevant, highly dominant as a source of financing, because in these early stages, when the risk of a startup is so high, there are very few companies that actually finance you, and venture capital. So you're not able to get an easy bank loan. So venture capital is sometimes the most relevant way of financing that. But if you go through the startup journey, actually in the end, you will use all of these measures of financing. And so did we at Adhesys Medical. So just to basically show you the same timeline here that I showed before with the pictures, just looking at the financing side. So when we started with Adhesys Medical in 2013, the first financing was through sort of friends, families, and the third F is for fools, so friends, family, and fools. So the very first investors and business angels who gave us 300,000 euros to just incorporate the company, get the patents into the company, and get started. Yeah, so that was our first round of financing. And then we continued for a couple of years with where venture capital was actually our main source of financing. So we did two larger venture capital rounds, one in Germany with German venture capital funds. A priced round is actually a round where you go to the Notary Public, you put a price tag on your company. That's what we did. We combined it with a so-called convertible loan, so a loan that can then later convert into shares of the company. And then once we opened our US office and through the connections of the RISE Business Plan competition, we found another group of US angel investors and US venture capital investors who invested into our company then again in 2015 and really helped us to go through a large part of the development. But we combined that financing actually with larger grants. And so we had in total 3 grants, 2 from the German government and 1 from the US government. And this actually helps you quite a bit to co-finance the venture capital money that you get in. And we use the grants not to finance sort of the critical path of the development of our technology, but rather used it to finance sort of all the side projects where we thought, okay, we need that. It's sort of relevant for us. It's a different application method. It's a different indication that we want to explore. And this we financed through these grant projects. And in the end, it helped us quite a lot because we were not the one-trick pony when we sold the company. We had different indications and product in the pipeline. So that actually helped us to create value. And then, of course, we also signed license agreements with the industry, which also helped us to finance the company in a later stage. And then eventually, once we sold the company, the development was not done. So we co-financed the continued development with the money then from the company that acquired us. So as I said, venture capital is sort of one of the most important aspects of financing a startup. And just to explain you the process of how to get venture capital. So the fundraising with VCs is actually a full-time job. So I was the CEO of the company. So for me, when we were in a fundraising phase, it actually took up a lot of my time. And so here's just an example. And I took this slide from another presentation from a VC firm actually about the different phases of the fundraising. So it starts with an initial introduction you get to a VC, and it's basically still true today that a so-called warm introduction where somebody knows somebody at the firm is very helpful. So as a CEO, you spend a lot of time traveling around the world and also going to conferences, meeting investors in person, and then sort of that you have a personal connect to a venture capital firm, or you know somebody who gives you an introduction to a venture capital firm. And then sort of the second phase is about sort of getting to know each other and exchanging information. So you send the executive summary or the pitch deck of your company. You have a first call. Then you still have meetings in person. Yes, this still happens. And sometimes multiple of those. And then sort of before you get the money, you actually then have to sign a contract. And this is actually more complex than it looks like here, because most of the time you first sign a so-called LOI, so letter of intent. And then there's a formal due diligence being done by the investor. Technical due diligence, legal due diligence, regulatory due diligence. So it takes some time and money. And then finally, you have a shareholder agreement with, like if you do it in Germany, you go to the notary public to get it signed. And then before the money is wired, actually, of course, you have to align with your existing shareholders. They have to be on board with that. And then you, as I said, you need to finally sign the deal at a notary public, and then you get the cash. So also a question that I get a lot of times about financing is, how do you actually value a startup? And this is a very difficult question. And from my experience now, also as an angel investor, you can do this in a very sophisticated way, and I will show you some of those. But in the early stage, honestly, it's pure magic. Yeah. So whatever you do, whatever sort of value you try to put at a company that has just been founded, will be wrong. Yeah. So it is basically a negotiation between the investor and you, and to really find out what is the price tag that you can logically put on the startup. But in later stage, it can become more sophisticated, and I will go into that. So at the beginning, it's the famous magic wand. So if you want to go a little bit more sophisticated, there are I would say there are 3 easy ways to do it. So one is the cash-in principle. So this means that you look at if you raise for a new investment round, you look at, okay, how much money has actually already gone into the company? Yeah, so you look at the previous investment rounds, the technological development, and then you say, okay, we add this much of new capital to the company, and so this is sort of the valuation that you need. The second method is to look at comparable exit figures. So you try to find companies that have been sold in the market, and you try to compare that to your company. And so those companies you find in the market, they will not be comparable. So they will have a different product. They have been sold in a different stage. But you will find this data, and then you have to deduct. Then you have to deduct the differences between your company and the companies you found, and this may help you to find sort of a value that is appropriate for your company. And the third possibility is then to do a so-called investment multiple assessment, where you not only look at sort of the exit values of the company, but also the how much money went into these companies and what was the multiple that this invested capital achieved. And from that, if you then again compare it to your company, you can come up with a valuation for your company at the current stage. And then in a negotiation, it really comes down to bringing the two sides together, a buyer and a seller. In the end, you're selling equity of your company, so you're a seller. The venture capital investor is the buyer. So you go into a negotiation about, okay, what is the right price range? And again, venture capital investors look at this really from a multiple perspective. So when they invest at a certain point of time, so for example, they invest at a valuation of 30 million, they will look at the potential that you have in an exit scenario. So if they believe that you can sell the company, let's say for 150 million, and now they're investing at 30 million, so that gives them an investment multiple of so-called 5x. So they can multiply their invested capital 5 times. And so this is what they are looking at, because they are in a high-risk business. So not all of the investments will turn out as successes. So they have to do a diversification of risk, but they also have to invest in companies that have the potential to also compensate other companies that may not yield a return to the investor. So I will not go into detail into that, but again, as a negotiation, you will think about the different scenarios, how you can develop your company, how also a certain exit can look like, and then you will find a negotiation sort of split in the middle with the investor. So that was a little run through about the financing part of the startup. Now I would like to talk about the steps to commercialize a life science technology. And I will do this a little bit alongside also the story of Adhesys Medical, which is a medical device company. Please keep that in mind. But I will also make some general remarks about pharmaceutical companies and biotech companies as well. So to start this, it's very important to differentiate really between the R&D phase, research and development, and then moving a product into the approval phase. Because as a scientist and also at university, you focus more on the early stage part, on the research and development, on basic research. And I really want to highlight there's a big difference between sort of this early R&D part and then moving a product into approval. And so I've seen a lot of great scientific breakthroughs actually that didn't make it to approval because there were a lot of mistakes made in the early phase, which then made it impossible to go through a realistic approval process. So the R&D phase is really about the orientations. You're exploring different technologies, different possibilities of doing this. And you can do R&D with a non-final material. So the material that you are testing with, that can be sourced basically from anywhere, doesn't need to adhere to medical standards, just also to use more cheaper material. But this is not possible in the approval process. I will come to that later. And then also, if you test your product, you will do, of course, first a lot of bench tests, but then in the R&D, it's very typical that you also test this with animals, which is the only way really of testing most pharmaceutical products and medical devices. But you start with the smallest possible species, which then is a mouse or a rat. And it may not give you a lot of insight then, of course, about how this works in a human. So this is really the start. But when you go to the approval process, things change. So the development needs to be done under so-called GLP, which is good laboratory practice, where you adhere to certain standards about the documentation of your experiments and so forth. So while in the R&D phase you can do a little bit whatever you want, in the approval process it needs to be all documented. And you need material from a validated production process. So this is a big difference. And you have to think about this already in the early stage. So sometimes the materials you are using in the early phase, you cannot get them in a medical grade. And this then causes a huge problem in the later stages. And then also in the approval phase, you have to do a lot of testing according to certain norms. I'm just mentioning here the biocompatibility norm ISO 10993, but there are a lot of other norms that you have to adhere to. So again, R&D, what does it mean? It means orientation. It means looking into new indications, exploring, testing, doing feasibility testing. And it happens under so-called non-GLP. So you don't have to worry too much about sort of the lab environment you're working in. You don't have to think too much about the documentation. Of course, you should document what you're doing, but you don't have to follow sort of a strict quality management system by doing that. But it's very clear that it's not sufficient for approval what you do. And many scientists make that mistake saying, okay, I'm done with the development, now I can just approve it. That's not the case. Because going through the approval process means something very different. It means to work with a final indication. It means that you cannot change the indication anymore. At some points, you will do a so-called design freeze, where you set the critical parameters of your product, and you decide on the indication that the product is used in, and then you cannot change it anymore unless you go all the way back and start over again. Yeah. And then what you have to do is you have to demonstrate at the same time safety and efficacy of the product. So safety, that is safe for humans, and efficacy means that it actually does what you claim that it should do. And then it needs to be done under GLP and under a quality management system. I'll come to that in a second, where you make sure that everything is carried out under a certain standard, it's monitored, it's recorded, it's archived, it's reported. Yeah, so this sounds really painful, and it is, but it's a necessity if you go to the competent authorities, notified body, or the FDA to get product approval. And again, you have to do certain tests according to certain norms. Again, as an example, ISO 10993. So what are the requirements for getting a product approved? And I'm splitting this into two sides. So there is the company side. What do you need to do as a company to be able to get a product approved? And then what you have to do on the product side, so on the individual product side. So as a company, the most relevant thing that you have to do is that you need to establish a quality management system. And that is different if you are doing medical devices or if you do pharma. There are certain, I'm just putting here some examples, but this is highly regulated. And actually the quality management system itself is monitored and approved by so-called competent authority. So you have you will be subject to so-called audits, where a regulator comes in and checks if you're actually doing what you're claiming to do on paper. So it's highly relevant for your company, and it's actually a no fault, I would say, no fault system. So where you may have to make sure that you always adhere to it. And there have been scandals in the past where companies have not adhered to this, and so it's actually a good thing that this is highly controlled and supervised. And actually, what does this quality management system do? It governs all product-related processes in the company and with the suppliers. So this is really relevant. So you're doing the quality management not only for your own company, what is happening in your own four walls, but you also have to monitor your suppliers, which then means you have to do certain agreements with your suppliers. You have quality documents that monitor the manufacturing of your product, for example. And then you also have to check. So you are doing audits with your suppliers to make sure that they are adhering to your standards as well. And then I've talked about documentation already a couple of times. It's one of the most important things. You have to ensure a flawless documentation of your development process, of the manufacturing process, and everything that your suppliers are doing. And this is actually also no fault situation where you really have to make sure that it's all correct. And what it does is that it allows you to have a consistent quality and safety of the product. So it's actually not just a burden for the company. You shouldn't see it like that, but it actually helps you that the quality and the safety of your product is ensured. On the product side, you also have to do a lot of things. So for a medical device, for example, you have to do a so-called conformity assessment to get CE approval. So in Europe, the all medical devices have a so-called CE approval under the medical device regulation. And so you have to go through this conformity assessment together with a notified body that checks that you adhere to all the standards and the requirements under the medical device regulation. For pharmaceutical products and biological products, there are different so-called competent authorities that are responsible for the supervision. So for medicines, it's the EMA, it's the European Medicines Agency, and there's also one for cell and gene therapy. So they will basically check if you are in line. And the same is also true for the US. So in the US, it's Food and Drug Administration, FDA, that will check if your product is in line. And as an input, there are a lot of norms out there that help you, from my perspective, to go through this process. It's the biocompatibility norm that I've mentioned before, but it's also guidance documents that the FDA or the EMA publish in order to basically help manufacturers to do the right steps necessary. And of course, you can look at what has your competitors done in the past. So you can look at competitor studies. What have they been doing in clinical studies, for example, to do the same thing. Right, so I will dive a little bit deeper into the key document now for medical devices. Just as an example, it looks a little bit different for pharmaceutical products, but sort of the principles is the same. The technical document is the main document you submit then for the approval of the product. It has 6 chapters. I will not go through all of them in detail, but it really covers all aspects of the product, from the characteristics of the product itself to the information about the supplier. Very important. As I said, you have to check them. You're responsible as the legal manufacturer of the device. You're responsible for the quality and safety of your manufacturers. So you audit them and you make sure that they are correct. Then, of course, design and manufacturing information for safety and for safety purposes. You put in all the documents that you have about preclinical studies, bench studies, and also clinical studies to make sure that the product is safe and has the efficacy it needs. Then also very important chapters about the benefit and risk analysis and the risk assessment of the product. So here, for scientists, it sometimes sounds a bit crazy, but you have to think about all eventualities, how your product can be misused. So if you use, for example, in our case, as a surgical adhesive, So we also have to look at, okay, what happens if the product gets into the bloodstream? Yeah, what happens if sort of it goes on organs or other surfaces that are not intended for the glue, and you have to think about what can go wrong, and have to provide a guidance what you actually do once it goes wrong. And so those are all the chapters you have to fulfill. In the end, a technical documentation can be thousands of pages, including all the studies you have done. So it's the key document for the product approval. And now, just to give you the example, in our case, for the surgical sealant, as an example, what this looked like. So as it was a class 3 device, medical device. You have a very simple description of what it is on a technical side. And then what is really, really important, and I said it before, that once you have chosen a certain indication, you cannot change it anymore. The intended use, so-called intended use, is highly relevant, which in our case was for vascular reconstruction and to achieve adjunctive hemostasis by mechanically sealing areas of leakage. That was the intended use of the product. And based on that, sort of you do everything else. And then also, just as an example, you provide all the steps of also how to prepare the product. For us, it was very simple because it's just basically a 2-chamber syringe, so ready to use. So you take it out of the packaging and then you just put it together and you're ready to go. But again, you have to show it, because from a risk perspective, what happens, for example, if the surgeon doesn't put on the mixing tip? Yeah. So what if he just puts in the glue like that? So those are all the risk aspects you have to look at. So I've talked about the documentation. I also wanted to talk a little bit about the product development and the different steps that are necessary. And here, this is also just an example of how this plan looks like. Yeah, and if you look at it, in the end, this will take multiple years to develop a medical device or a pharmaceutical product. But you will go through the same stages, of course. You need to first of all, you need to do all the R&D, which is not here actually on this chart at all. Yeah, but then you have to establish and finalize the manufacturing process. As I said, to have a final product design, you need to be able to produce so-called GLP supplies. You can then do a feasibility study and other studies that are relevant, like a long-term study that we did in this case. And also down below there is the so-called CV trial, which was then a clinical trial of the product. And so you have to go through all these stages. And what is also relevant, you see here, there is the FDA interaction. I will highlight this here. So you also regularly talk with the competent authorities, which in our case was the FDA in the US. So you get their input. The FDA they're not sort of giving you advice what you should do. They are rather saying, okay, we want to see this and that. But it's really important that along the development process that you have a regular interaction so that you know actually what the FDA is expecting from you. And here is an example of the so-called bench testing or in vitro testing that you have to go through in our case as a medical device, which are relevant. So bench or in vitro it's not animal testing, so you do this in a lab. And what is really relevant for all medical devices is the so-called shelf life. So how long can you store a product once it's done with manufacturing? And so the target is at least 12 months, for example. But most devices aim for a longer period than that. And then for us as a surgical adhesive, it was really relevant to show the adhesive strength and the elasticity. So we did multiple tests about that. And then another big bucket of testing was about the degradation. So how does the product degrade in the human body? And so we did a lot of testing about that. So actually degradation is very difficult to show because if you do a clinical study, you cannot you do surgery on a real human, so you cannot open the person up again just to look if the product is still there. So so you can do that in animal testing, but in humans you cannot do it. So we had to do a series of testing on a chemical side and on a theoretical side to show that this is actually working, because the final proof was of course not possible to bring in a human in a clinical study. And then another big bucket is the safety and performance testing. So safety and performance for us had two sides. So one was we did a very comprehensive paper-based risk assessment. So we had external parties, external research laboratories doing an assessment of the product regarding the degradation again, and also the biological risk. And here, sort of based on the chemistry of the product, these institutions did an assessment if it is harmful for the human body. And then we did a huge series of ex vivo and so-called in vivo studies to further test the product in animal studies and also in other bench studies, to show that the product works and it's safe and it fulfills all the criteria that we are claiming. And then the next bucket of testing is then sort of the biocompatibility testing and GLP testing. And here, as I said before, I mentioned the norm 10993. It's sort of a very important guiding norm for medical devices. It includes all these different aspects of biocompatibility. And actually, the good thing about this norm is that you already know what you have to do. So you can look it up on the internet, you can Google it, and in the different chapters, you find the different tests that you can do. And so actually, it helps you, guide you through the process of finding out what you need to do for your specific device. So that was actually all I wanted to show you about the preparation of a startup. Of course, as I said, it has multiple dimensions to build a company, and I just focused on the financing part and for life science on the part of getting the product through the approval process. But of course, it has other aspects of sort of also building the organization, building a team, and also building a organization that can license the product or commercialize the product. So I've not touched upon this at all, but just to give you an idea of what it takes to bring a product to the market. Now, in the last part, I want to talk a little bit more about a fun aspect, I would say, about building a startup, because it's also something that I learned when I started Adhesys Medical, which was my first startup, that actually when you build your company, the hardest part is also to convince others to sort of support you. And support can be in many ways. It can be giving you finance. So venture capital investors, they want to see a famous pitch deck. Yeah, they want to see a pitch deck from you, but also convincing your first employees, because you're a startup, you're a small company, you don't have a lot of money. And so I found myself in the situation that I was pitching the company and the idea to people on a daily basis. And I was actually not prepared for this pitching part, because also I came from university, I came from academia, I was doing my PhD, so I was very much in the more in the explaining way. I was always trying to explain what we are doing. I was not pitching, and this is what I want to talk a little bit about right now, what pitching is and why you should learn about it. And so pitching is selling your big idea to the investor, to your next employee, to even your friends and family. Yeah, you also have to convince your parents that it's a good idea to start a startup and not, pursue another career. So it's selling your big idea. And to compare pitching to presenting, or sort of this technical analysis, is like if you would compare writing a doctoral dissertation about the chemistry of affection compared to Elvis Presley's song "Love Me Tender." Yeah, so it is a completely different approach, maybe about the same thing, but it's very different in the way of doing it. So most of you have already done a presentation, and so if you want to learn more about pitching, I think you can relate to some of these topics that I will show you. So if you have ever done a presentation, have you ever felt like that instead of looking at the big picture, the audience just kept questioning your numbers, so they focused on the nitty-gritty stuff, or you had to repeat the key arguments over and over again, and still nobody would get them, or the people seemed to be interested, but nobody really caught fire. You were so on fire about this subject, but nobody else was really showing a reaction. Or you overwhelmed, maybe also self-critically, overwhelmed your audience with facts and details and figures, and nobody, afterwards, nobody even dared to ask a question because, you were just overwhelming them with facts. And then you explained the science about your product for more than 20 minutes, and then somebody, and you think that you were really convincing, but still somebody said, okay, this will never work. Or instead of asking intelligent questions, people just pointed out that there was a typo on slide 3. Yeah, a very typical thing happened to me a lot of times. So that happens to all of us, and the problem is really the human brain. So how can that be? So our brain is actually not made for processing PowerPoint presentations or following PowerPoint presentations. And I'm talking here to mostly scientists, I assume, so you know this better than me, but there are the 3 parts of the brain: the neocortex, the limbic brain, the brain stem. And so the neocortex is actually where we create presentation. It's the part of the brain that is responsible for the so-called higher order thinking, where all the logical logical information and data is processed. But this is the brainstem, which is actually sort of the oldest part of the brain that is responsible for our survival. So about breathing and sort of giving the impulse to run away if there is sort of a tiger somewhere. This is where we actually receive the presentation, and then And this actually is a problem. So if you think about it, so while you are presenting sort of all this information and these slides and graphs and whatnot, actually, if the people are receiving it with their brainstem, they are thinking about, running to the bathroom, so primary needs, or they're maybe hungry, or they just want to get the hell out of there. So this is how actually you have to think about your audience. They are not prepared for what you're showing them. It is not our fault. Actually, the logical thinking is very new to humans. So if you look at sort of the evolution of humans in the time span, so from the very first creatures to the first, what we call in the family of humans, the Homo erectus, 2 million years ago. Actually, the Homo sapiens, the human as we sort of know it, came only into being a mere 200,000 years ago. So this is very, very new. But if you look at the long time span, it's actually quite interesting that it took the Homo sapiens just 100,000 years to conquer the entire world and to really develop as a species. And actually, what was the trigger for that was the so-called cognitive revolution, which allowed humans to do all these things. So we moved from a state where we couldn't, communicate or couldn't process higher order thinking. We moved to a stage where we were actually able to convey complex messages to others. Yeah. So I've seen a lion at the lake. So not long ago. So better you run. Yeah. So it actually helps with survival. Yeah. And also, we were able to specialize and do teamwork. So we moved from a situation where all men hunt to a situation where people were specializing somewhere, building weapons, or somewhere growing food, providing food. So that was a big step. And then also, we were able to draw conclusions and make complex plans, like from moving around and hunting, we came to the phase where we were actually, able to say, okay, why don't we stay here and build a real house and grow food and keep goats. And so that happened over the last 100,000 years, and our brain is sort of slowly adapting to this. And if you want to transpose this now into conveying a message in PowerPoint presentation, actually you have to address all three parts of the brain. Yeah, so the neocortex, the brainstem, and also the limbic brain. And the limbic brain is responsible for emotions. And also, if you listen to presentations, if there's no emotion that catches you, you're not interested. You will doze off. You will play with your cell phone after a few minutes. So the first step is to really address the emotions of the audience. You have to make sure that they understand that this is relevant. So in life science, it's always about, it's saving human lives. And if you remember my presentation, I was talking about sort of the human catastrophe of bleeding related complications. This is where I try to create a frame for you that you say, wow, half of the surgeries we still have a bleeding related complications. That's more than I expected. That creates a frame that draws an emotion. And then you have to keep the attention. So again, the brainstem will tell you to run away. If you want to avoid that, through your presentation, you need to keep the audience attention. You need to keep them interested. So you have to have a good flow within your presentation. And then once you have that, you can address the neocortex. You can address the higher order thinking, and you can convey a message. So you have to make sure that both the limbic brain and the brainstem, they are taken care of with a frame and sort of with a good pace in the presentation. And then the information can come across, and then you can deliver the numbers. So just to sum up how to do a successful pitch, create a frame, which means owning the situation, give the example that catches the emotion and the attention of the people. Once you have the attention, keep it, create tension throughout the presentation, keep a good pace. Have interesting information that is coming with every slide, that the people stay focused. And then lastly, move towards a decision. If you want to convey a message that results into action, you have to do it in a way where people say, okay, yeah, now I have to do something. Now I'm convinced, I've understood the numbers, I want to do something. Always close sort of with a call to action to your audience. And so this is all the theory. If you want to go a little bit into practice after this, I encourage you to Google some great examples of pitches of RWTH startups that have presented also at the RISE Business Plan competition that I mentioned before. So you can go on YouTube. I put the links here. You can type them or you can just Google them. LydroTech and FiberCode are the names of the startups. Those are long videos, so I also provided the timestamps here. You can scroll to the timestamps and watch the presentation, and you can see how they are doing it, how they're delivering a good pitch. For me, they are great examples of how they did it. Actually, LydroTech also won the RISE Business Plan competition, and FiberCode placed third. So, and if you do, challenge yourself. If you watch it, you can ask yourself the following 5 questions and put yourself into the shoes of a venture capital investor. And venture capital investors, they base basically their decision on those 5 questions from my perspective. So first of all, think about the market. Describe the market that the startup seeks to address, and think about, is the market big enough? Is it an attractive market? Then think about the problem the startup is solving. Is it a relevant problem? And third, do they have a solution that is actually can make a difference here? And then also relevant for deep tech startups, do they have the right technology? Is the technology differentiated from existing solutions? Do they have a unique selling proposition? And then lastly, do they have the intellectual property or some other sort of technological advantage to defend their position, so which is about the intellectual property. So go to YouTube, look at the videos and see some good pitches. And then my last call to actions before I'm going to close this lecture. So if you're now interested about entrepreneurship, there are a lot of institutions, especially also in the scientific world, that you can go to, check them out. If you're a student at RWTH, there's the RWTH Innovation, which has a lot of offers. And also within Germany, there's the Unternehmertum, who are doing this in Munich. And then also research organizations like Fraunhofer or Max Planck, who are providing support for scientific entrepreneurs. If you're looking for co-founders, so if you yourself, you have a certain specialization, if you're looking for other people who might be who should join your team because you need other skills in your team, you can find those also on different platforms I'm showing here. And my last recommendation, just to get a taste of entrepreneurship, I encourage you to visit an event that is focused on entrepreneurship and maybe especially on deep tech. So check out any of those just get a feeling of entrepreneurship, and maybe it is for you a good alternative to make your science come to reality. And with that, I would like to close. Again, I'm really happy that you followed my lecture to this point. Please connect with me on LinkedIn. Send me a message if you like that presentation. I'm very happy for everybody who is considering making a move and making scientific research a reality. And if I can support in any way, I'm happy to do that. And with that, I would like to close. Thank you very much for your attention and all the best for your future. Thank you.